So translation is where we're going to be taking that, that messenger RNA transcript and using it as a template to create a protein product. Okay, so this time the template is messenger RNA specifically. Okay, so I think we made a comment of it's RNA, but very specifically the template is messenger RNA. And here's where we are going to be having, so the messenger RNA is going to serve as the template. Here we have this is actually an organelle, the ribosome, and the ribosome has RNA in it called ribosomal RNA as part of its structure. It also has proteins in it, so it's a combination of proteins and ribosomal RNA that create that ribosome. The ribosome actually consists of a small subunit and a large subunit. It is that are actually kept separate. They don't interact together unless they are interacting with messenger RNA, the template. Okay, so they are separate until they're on that messenger RNA. And then, of course, when they do come together on that messenger RNA, they are going to create three very specific regions within that ribosome. We have the A site. Okay, the A site is for the acetyl tRNA binding site. So this is where the tRNA with the amino acid is going to be able to come in and interact with the messenger RNA. We have the P site, which is the peptidyl tRNA binding site. This is where the growing polypeptide is going to be found attached to a transfer RNA. And then we have the E site, which is the exit site. Okay, and so this is where just the transfer RNA that no longer has an amino acid attached to it is going to leave. Okay. Actually, is this one's flipped. Based on, they're going the reverse. I don't like this figure. They're kind of going the reverse of what, you're going the reverse. So in this case, you see the amino acid. Usually, figures I see is it's the A, P, and E, and they're going in that direction. This one apparently shows the amino acid bound here. So that would be the A, this is the P, and the E, based on this figure picture. All right. Still, what's in those locations is the same, right? A site is for the incoming amino acid. The transfer RNA that has the incoming amino acid. The P site is for the growing polypeptide, and the E site is where the transfer RNA can leave. I really don't like this picture. So the small ribosomal subunit is actually going to interact to a ribosomal binding site region on the 5' end of the messenger RNA. And then it's going to scan that messenger RNA until it reaches the first AUG codon. Okay? The AUG codon would be where we see that those nucleotides, A, U, and G, present in that order. Okay, and that is going to bring, then it's going to pause in that location, and the transfer RNA carrying a methionine is going to sit in that location. So it's not like what you see here at the moment, but when you're initiating, it'll sit in the P site. That is what sets up the reading frame for continued transcription, displaying then the next codon in the A site. Okay? All right, so messenger RNA, its function is it serves as the template. Okay, that's the key one that we were really kind of talking about when we were talking about transcription. The other RNAs, ribosomal RNA and transfer RNA, are also transcribed the same way, but there's not as many of them transcribed. The most products are going to be messenger RNA. Okay. transfer RNA in this process is what's going to actually bring in the next amino acid. So it kind of it is kind of the shuttle, as well as the ability to recognize what the next amino acid is supposed to be, because the transfer RNA is going to have an anticodon that can base pair with the codon. All right, so that anticodon is going to base pair with the codon at the A site, bringing with it on the 3' end of the transfer RNA is going to bring with it the next amino acid, so the related amino acid. Essentially, since we have we have the 20 amino acids, we have that many transfer RNAs, one for each of the different amino acids. And as we look at the codon table in a little bit, in order to be able to translate a messenger RNA sequence based upon the codon, right, we're going to find that there are going to be several transfer RNAs that are going to recognize different codons. Okay, so there's going to be transfer RNAs that can recognize more than one codon, and that is due to the wobble position, the third position of the codon that's going to recognize is not as tight of a base pairing interaction as the other ones. Okay, and therefore we find that multiple codons can give us the same amino acid. Okay, that then gives us, when we look at the codon table, what we call, it depends upon the description. It's either referred to, I've heard it referred to as the redundancy of the codon or the degeneracy of the codon. Okay, the fact that one codon can give us the same, more than one codon can give us the same one amino acid. The transfer RNA does have a 3D structure. The 3D structure, when flattened out, has a clover leaf appearance to it with these 3 hairpin loops. Okay, and but visually more in that 3D structure in a more laid out format as what we see in the cell. It's going to be looking like this, where the anticodon is one end and the amino acid binding site is on the other. A transfer RNA, can be reloaded with a new amino acid, so it can be reused over and over again. Okay, so it gets loaded with amino acid, plays a role in translation, drops off that amino acid in that process, and then is able to go back and, once it exits the ribosome, get reloaded with a new amino acid. If we look at the structure and function of the 3 main types of messenger RNA, the 3 main types of RNA, messenger RNA is going to be the one that serves as the template, okay, and it is what is going to be translated to give you your protein. Ribosomal RNA, there's several different ribosomal RNAs. They're all going to play in a role in producing or assembling that ribosome. In a prokaryotic cell, the ribosome is going to be assembled in the cytoplasm. In a eukaryotic cell, those ribosomal subunits are actually assembled in the nucleolus that is found within the nucleus. Okay, and then the subunits are then transported out into the cytoplasm. Transfer RNA, okay, is there's going to be multiple of these, one for each of the 20 amino acids. Okay, and they are what's going to carry that amino acid into that reaction based upon the codon and the fact that they're going to have the anticodon that is going to recognize the codon on the messenger RNA. The codon table is as follows. Fortunately, you do not have to memorize this. This is something that would be provided, right? But codon table is as follows, is set up as follows. You're going to, when you have a sequence, you read 5 prime to 3 prime, and your first letter of that sequence is going to be, that codon is going to be found on the left-hand side. Then your second letter on the top, okay, so if we have CCA, I would be looking for C, something along here, right? C is my second, so I'm in this box. Usually by the time you get to the third, you're already in a square, it's pretty easy to locate. However, they do tell you the third letter is on the right, is listed on the right-hand side, okay, as to where it's located in that box. You'll, as I mentioned before, you'll notice that multiple codons you can find throughout this codon table, multiple codons will give you the same amino acid. So these 4 give you proline, okay? And that has to do, you'll notice that the first position and the second position are the same. It's the third position. That's the wobble position for the transfer RNA. The wobble position is the one that has less concise base pairing, okay, so that any one of these nucleotides in that third position is still going to be interacting with that same transfer RNA that has protein. Okay, there are, based upon the fact that the codon consists of 3 nucleotides, right? That means we have about 64 options out there of codons. We only have 20 amino acids. Okay, so more than one codon is going to give us the same amino acid. Later, when we talk about mutations, this is going to play sometimes to our benefit when we talk about, say, silent mutations, where a change, say in the position of where we would have had the third nucleotide for that particular codon, it might not actually result in a change in amino acid. You'll notice also on this table, we do have the start is for the most part, most of your proteins will start with AUG, okay? And that codes for an amino acid, methionine. All right, so that'll be the first of the amino acids. And then from there, and that sets up the reading frame. I have a little bit, we'll talk about the reading frame itself in a little bit when I have a figure to place that on the screen. But then from that point, every 3 is being read until you get to one of the stops. Once you hit one of the stop codons, the process ends, okay? And the ribosome will fall off, and as well as the polypeptide, of course, will be released. The stop is not an amino acid. It is there are release factors that will then sit in the A binding site where when one of those 3 codons is displayed. Okay? All right, so the process itself has those 3 stages: initiation, elongation, and termination. So initiation is going to start with the small subunit interacting with the messenger RNA, and it is going to locate that, so it's going to bind near the 5' end of the sorry, I'm going back to that figure I just said I didn't like because it was where the 5' and the 3' were located, and they have it backwards. The picture is backwards, but regardless, it's interrupting me, it's irritating me. But anyways, so the small subunit is going to bind to the 5' end of the messenger RNA and scan until it hits the AUG. The transfer RNA carrying that methionine is going to be is going to interact at that location. We will use GTP as our energy source for this for translation, and a GTP with the use of GTP, we will bring in our large subunit and we will get our completed structure That is the complete ribosome, small subunit, large subunit, and interacting together with the messenger RNA running between. Okay, and our A site, our P site, and our exit site, okay, are going to be in position. The AUG, so the transfer RNA with the methionine sitting on the AUG, interacting with that AUG site, lines you up, notice, with our A site now is displaying the next 3 codons. So the transfer RNA that can recognize it, we have now had initiation. This is the end initiation. We move into elongation. The next transfer RNA, who has an anticodon that can base pair, will base pair, bringing with it the next amino acid that's being requested. The ribosome will catalyze that peptide bond, and so that growing polypeptide, it gets transferred over to that new incoming, the transfer RNA that just came in. And then we will have translocation. The ribosome will shift, moving the position so that once the bond has been made, now our polypeptide sits here. The ribosome will shift so that that polypeptide now sits in the P site, displaying the next codon. And that repeats and repeats and repeats. Each time we're using GTP as our energy source. Okay? It will continue to repeat down that messenger RNA until it reaches the stop codon, one of those 3. Okay? When that stop codon sits in the A site, we have what we call a release factor that's going to then interact with that stop codon. The ribosome is going to catalyze the reaction, right, that of peptide bond formation, but there's nothing to attach it to, so the growing polypeptide is released. As soon as it's released, the 2 subunits of the ribosome will fall off the messenger RNA. And we've ended our story of translation. Okay? Let's see here. The polypeptide itself will, while it's being formed, will create those secondary and tertiary interactions. And we also know that there are some proteins that can be involved in that process too, of making that 3D structure with the hydrogen bonds of the secondary level of structure and the hydrogen ionic, hydrophobic, and disulfide bridges, which are covalent bonds within the tertiary structure. This, what you see here occurring with the translation, this process is where we're actually making the primary structure, the amino acid sequence that are connected by peptide bonds. Comparison of translation in our bacteria versus eukaryotes. So our prokaryotes versus our eukaryotes. Well, to start off with, we already know the ribosomes are different. Okay, we've actually covered that in the first unit. The ribosomes for the bacterial cells are the 70S as compared to the eukaryotic cells. It's a larger ribosome, and it has to do with the proteins and the ribosomal RNA that are found in those structures. Regardless of the difference in size, the way they work is conserved. The way that the overall structure and the fact that they have the A site, the P site, and the E site still exists. So that's still there. Okay, they're just one's a little larger than the other one due to the particular proteins involved. The initiator transfer RNA. In bacteria, it's a modified methionine as compared to eukaryotic cells, it's just a straight methionine. Okay. I'm not going to go into this. Simultaneous transcription and translation. You can have that occur in the bacterial cells because of the DNA being in the cytoplasm. You cannot have, so this is what we had talked about right before the break. You don't see that in eukaryotic cells because transcription occurs in the nucleus, translation occurs in the cytoplasm. So they can't happen at the same time for a eukaryotic cell. After translation, regardless of a prokaryotic or eukaryotic cell, after translation, these polypeptides can be modified by having, say, a carbohydrate added to it or a lipid added to it. The post-modification is possible in either bacteria or eukaryotic cells. For eukaryotic cells, if it is protein that's going to be found in the nucleus, then we see it made by free ribosomes if it's a protein that's going to be made in the cytoplasm and stay in the cytoplasm, I don't know why nucleus came into my mind, then it's going to be made by free ribosomes in the cytoplasm. If it is a protein that's destined for an organelle, for a membrane, or to be secreted, any of those, then we will see the endoplasmic reticulum come into play. The ribosomes are not attached to the endoplasmic reticulum indefinitely. What happens is, in a eukaryotic cell, is that free ribosomes attach to the messenger RNA. They start that translation process. A signal sequence is immediately, as part of the beginning of that process, is translated, and then there are proteins that recognize the signal sequence, and they will bring that ribosome to the endoplasmic reticulum and help in the attachment, so that as the ribosome continues to translate, the polypeptide is produced into the endoplasmic reticulum. Okay, and then from there it goes by the vesicles over to the Golgi apparatus, and then that's oftentimes the modifications occur in the Golgi apparatus and get shipped to wherever. Those are a lot of extra steps that a eukaryotic cell has because of its compartmentalization. Okay, a prokaryotic cell, it's all free ribosomes, happening in the cytoplasm. Okay. All right. I think that covered everything that I wanted to cover in this one. All right. So how do we interpret, say, a nucleotide sequence or read a nucleotide sequence? First of all, I want to bring up, so I've got a sequence, 5' to 3'. The ribosome attaches to the 5' end and scans towards the 3' end. So the small ribosome attaches first at the ribosomal binding site and then starts the scan, and then it will stop at the first AUG. Okay, so if the small ribosome is going to start moving in this direction, the first AUG is right here. That AUG is going to give us a methionine. Okay? So based on that, my next is AGC, which is going to give me what? SER. Okay, serine. Okay. GGU, I'm okay with you just giving me the 3-letter.
Gly.
Yeah, so you said line.
GLY.
Oh, glycine, okay. UGU. Cysteine, okay. UAA. Stop. Okay, so this is not, that's not an amino acid. Okay, So if a ribosome were to read this, that's how the reading frame would be set up. Now I say reading frame, and I understand you guys have had some you have had this covered before in general biology, but I wanted to show you the other quote unquote reading frames that could exist if we didn't know where to start. Where did I put my pen? We did this, right? So this was R3, and it continued like that. And there, and there. Okay, but what if I started here? What would my amino acid sequence be? I'll get out of your way for this. CAU. Hint this, okay. GAG. Glue. Okay. CGG. Which one? Arg. Arginine. UUG. UUG is leucine, okay, and UUA, also same, right? So the amino acid sequence is different. But that was if we started there. What if we had started here? We're going to end up with a whole nother potential sequence of ACA, threonine, okay, UGA, let's put it at UGA. That's already a stop. Okay.
So how does it know where to start? Is it that
No.
bad?
Thus the importance of the ribosome identifying the first AUG on that transcript, because that sets up the reading frame. Because you have a codon consisting of 3 different letters, you need to know where we're going to actually start. With 3 nucleotides playing a role in our codon, you have a potential of 3 different reading frames, which clearly give you 3 very different outcomes depending upon where you start. Okay, so the whole purpose of that start codon, the AUG, is to establish that reading frame so that you know none of those are correct. The only answer to this is listed there.
So basically we have to always start with the AUG
Now,
and read from there.
here's the catch, of course. You have a multiple choice test. And therefore, on the multiple choice test, you'll start at the beginning of whatever I have listed. Okay? And no, you won't be looking for the AUG. In the test, the first codon, the first set of 3 is your first codon. And quite frankly, because it's a multiple choice, only one of them is going to match up correctly anyways, right? I mean, it's not like I'm asking you to do this. It'll be, you will do, I will be asking you to do it, but you won't be doing it as in handwriting your answers. You'll have your different possible, and only one of them will match. So on a test, you would start at the beginning and read through. Okay, but reality is they don't start at the beginning. The ribosome does not start that first amino acid. In fact, if for a messenger RNA transcript, there's actually a pretty lengthy area that is not read and that's not translated at all. You have your RNA binding, your ribosome binding site up there in the front near the 5'. That's not translated. And then it's going to scan for a bit until it finds that first AUG. So when we say that our messenger RNA transcript, right? So our messenger RNA transcript we want to know how long our messenger RNA transcript is for our protein. Okay, the protein it produces is say 10 amino acids. How long minimum, how long does that messenger RNA have to be in length, in nucleotides? How many minimum nucleotides must it have?
10.
30.
30. 30, because every amino acid consists of 3 nucleotides.
Oh.
That's the minimum. Reality is this messenger RNA transcript might be more along the lines of 100. All the others, nucleotides are not involved, or not in that, are not going to be translated, okay, in this process. Now, this is this protein that's produced. It does have direction to it. We have the C terminus and the N terminus. This will be the C terminus. No. This is the N terminus, excuse me, and this is the C terminus. Okay, of a protein, because don't forget, proteins have a backbone. Their backbone is NCC, NCC. It repeats, NCC, over and over again. The N from the amino group, the C from the central carbon, and the C from the carboxyl. Then followed by the N for the next amino acid, the N from the amino group, the C from the central carbon, and the C from the carboxyl. So it repeats, so that it is directional. The differences, remember, for those amino acids, the difference is not the backbone. That's all the same. What makes it unique is then the R groups, and so the order of the amino acids, because the R group of a methionine is different from a serine, is different from a glycine, is different from a cysteine, right? Those R groups are how we see differences in, and the sequence of the R groups that were being displayed are what's going to make that protein unique. Did I determine polypeptide? Okay, sure, that answered that learning goal. Right? All right, anything else on this one? All right, so now we move into regulation of these processes, okay? So when we talked about the proteins themselves during metabolism, we said that they were regulated, right? We had inhibitors and activators. They're going to be controlled by the temperature and the pH. The activity will increase up to a certain point with the presence of the substrate, but at some point the substrate there's a saturation point, and so, we see a plateau occur. We talked about all of that. In regards to then protein control. The protein's already produced. Now we're controlling the protein on whether it's functioning or non-functional, right? Active or non-active. But there's also other ways in which we may control the protein that has actually nothing to do with the protein itself, but rather steps before the protein is produced. So if you do a control through the protein itself, that means the cell has already expended energy. It's already gone transcription, translation to create the protein. The protein's there. Now we're turning it on and off, right? And that's beneficial for some types of metabolism. But other types of metabolism, that's actually a waste of energy. Why are we going to bake the protein if the substrate isn't even available, right? If the substrate's not available, I don't want, as a cell that's highly efficient with its energy source, because it's a limited energy, so it needs to be efficient with its energy, the cell's going to say, I'm not making the protein then. Substrate's not there. If I make that enzyme, it's not going to get used, so that would be a waste, so I'm not going to make it. Now, that type of control is going to be it can occur either on the transcriptional level, so we're going to control whether transcription occurs or not, or it can be on the translational control level. Okay, how can we control on a transcriptional level? The promoter plays a big role in that. Remember those two key points, the top, so the -10 and the -35? The closer they are to the consensus sequence, the more likely that promoter is going to be recognized. The more they vary away from the consensus sequence, the less likely it's going to be recognized. Okay? Will it get recognized? Sure, but not as frequently. Other things that for transcriptional. You remember the double helix? There, in the double helix, there's what we call a major groove and a minor groove. The major groove it exposes more of the DNA to proteins. Minor groove is a smaller distance, so it's less accessible. So if the promoter is located in the major groove, it's more accessible. If the promoter is located in the minor groove, it's less accessible. We will also be talking about in the next couple slides a type of transcriptional control in regards to the prokaryotic cells specifically with the operon. The operon is a transcriptional control where it will actually shut down a section of genes by a regulatory protein. And only, so the protein interacts with the DNA, preventing transcription, and only when the substrate is present do we see that protein disappear, okay, or be removed. It doesn't disappear, it just gets removed from the DNA. Another thing that what eukaryotic cells do is a methylation process. We see this, you guys would have talked about in general biology when they were identifying dad's genes and dad's DNA versus mom's DNA. So in the gametes, all DNA, right, is going to go through a methylation process that designates it basically. That certain genes are going to be methylated, or certain areas of the DNA are methylated, shutting them down, so that those particular genes are more likely you're going to get from your dad or your mom because of the fact that methylation exists. So those are types of transcriptional control that's out there. Translational control would be the messenger where transcription occurs, but then when it comes to translation, it's blocked in some way or other. This is used by the cell, but less frequently used because now you've still expended the energy to make the messenger RNA. Okay, so if you want to stop a process with the least amount of energy, you don't go halfway. You stop right at the beginning. Okay, so transcriptional control is the preferable way as compared to translational control. Translational control is through things such as our regulatory RNA that will interact with the messenger RNA, making it double-stranded, and therefore the ribosome can't bind to it. Okay, so those are things that can happen with the translational control. For the transcriptional control, though, in prokaryotic cells, they have a unique gene structure called the operon. The operon is where they set a group of structural genes. So in this case, A, B, C, and D. Those are 4 different genes to produce 4 different proteins, but they're all back to back under one promoter. Okay, so these 4 genes are controlled by one promoter, and those 4 genes are usually involved in the same metabolic activity. If I need to do this metabolic activity, metabolism is a step-by-step process. I need these 4 genes. Instead of individually producing them, right, the prokaryotic cell, this doesn't happen in the eukaryotic, by the way, again, stress, not in the eukaryotic, not in our cells, not in eukaryotic cells. In the bacterial cell, it will put it under the same promoter. Thereby, once you recognize that promoter, you get all the enzymes in one shot. This speeds up another way in which the prokaryotic cell can speed up its production of proteins for a given metabolic process. Now, in addition, though, you'll notice there's something else. Between the promoter and the structural genes is the operator. The operator controls whether these structural genes are going to get transcribed or not. Okay? If something is bound to, if protein is bound to this location, then even if the RNA polymerase can recognize the promoter, it can't move forward. So it'll never transcribe these genes. If the protein, regulatory protein is not bound, then when the RNA polymerase recognizes the promoter, we get the full transcript, messenger RNA transcript, which then can be translated into the 4 proteins. Each of these genes will, on the messenger RNA, it'll have a ribosomal binding site for each of the 4 different genes. Okay? So, how do we control the operator? Well, the operator is controlled by that regulatory protein, which is produced by the regulatory gene. This regulatory gene, I don't like the coloring on here, has its own promoter, okay? And then the structural gene. This, when it is, the RNA polymerase recognizes the promoter, it produces that product, so a messenger RNA is produced, protein is produced, that's the regulatory protein. Now, the regulatory protein, it depends upon the particular operon as to whether it is going to repress the operon or induce the operon. Inducible operons are usually linked to catabolic processes. Inducible operons are ones that are normally turned off unless the substrate is present, which interacts with the regulatory protein, preventing it from binding the operator, and we get production. Okay, so when the substrate is present, the operon is induced to produce the protein products. And usually those protein products have to do with metabolism of that substrate. So the proteins are only produced if the substrate is present. A repressible operon is one which is normally turned on, but sometimes the cell needs to shut it off for a little bit. That's usually linked to an anabolic process. Okay, so it's similar to a feedback inhibition, but not on the protein level, but rather transcriptional control level. Usually linked with where the end product is going to interact with the regulatory molecule, and when it does, when the end product and the regulatory molecule interact, that binds the operator, and we no longer get our messenger RNA. Okay, so that is the 2 types of transcriptional control. Okay, that we can find. Are all the genes set up like this? No. Okay, again, this is in the prokaryotic cell, and it is not all genes. It's only some pathways that might have this operon set up. 2 that we're going to look at. We're going to look at the TRP operon and the LAC operon. The TRP operon is a repressible operon. What that means is it's normally turned on, and occasionally it gets turned off. Okay? So when the regulatory gene is transcribed, it produces the repressor, and the repressor is, when produced, is an inactive state. It is in a 3D configuration that does not allow it to bind to the operator. As a result, since nothing is bound to the operator, it can't bind here. When RNA polymerase recognizes this promoter, it can transcribe our genes, and we're going to get protein products. That metabolic pathway is going to proceed. Now, when tryptophan becomes, it's the level of tryptophan increases in the cell. It's an amino acid, but it's not getting incorporated into the product is not getting incorporated into, say, whatever protein synthesis the cell is doing fast enough. So we're seeing a buildup of tryptophan. Well, tryptophan will interact with the repressor, and when it binds to an allosteric site of the repressor now can bind the operator. If RNA polymerase recognizes the promoter, the repressor's in the way. So it can't move forward. We don't see transcription of those structural genes. All of these genes have to do with that metabolic process of making tryptophan. So we're going to see a decline in the amount of tryptophan produced. And you say, well, don't we already have the enzymes out there to make tryptophan? Yes, the cell does. But proteins that the cell has already made, they have like a lifespan. They do cycle, right? And so as they cycle out, right, then we're going to see that slow decrease of tryptophan. Tryptophan, of course, is being used to make other proteins, and the levels are going to start to drop. The interaction, once the levels drop low enough, the interaction between the repressor and this tryptophan are going to separate. It's reversible, so it's going to separate. Repressor now is going to be we will no longer be able to interact with the operator, and we now have the operator turned back on. Okay? So a repressible operon is normally on, occasionally turned off. Okay? Think about it as light switches. In your house, you probably have that room, like the family room or the kitchen or the great room, whatever you want to call it, a combination of the two, where the lights are almost always on. Middle of the day, and the friggin' lights are always on, right? But occasionally they get turned off. That's what's going on here, as compared to an inducible operon. An inducible operon is more like the bathroom lights. It's turned off, and only when you need to go in to use the bathroom do we turn it on. Okay? Here we have the lac operon example, and the lac operon consists of a promoter, the operator, and structural genes, all having to do with the metabolism of lactose. So if lactose, in this case, when the regulatory gene, that is in another location, anywhere in the chromosome. It doesn't have to be near the operon. That is what we consider a constitutive gene. It's not under any sort of control. It's always being made. And so we'll have that repressor being made by the cell. The repressor, though, when it's made, is in an active state so that it can bind to the operator. And therefore, RNA polymerase will not be able to transcribe these structural genes because the repressor is in their way. When lactose is present, when that substrate is present, the lactose is actually a derivative of lactose, but it will interact with the repressor. And that interaction will make that repressor inactive, so it will fall off of the operator. Now RNA polymerase can transcribe the genes, produce the protein products to metabolize lactose. As lactose depletes, this interaction ends, repressor binds back to the operator, and we stop making those genes. Or we stop transcribing those genes, we stop making those proteins. Okay? Now, just to throw a loop into this whole thing, does that make sense? So this one's turned off normally, but only when lactose is present do we turn it back on. Okay? Now, there is, though, a caveat with this whole lac operon, and that is the preferential treatment that bacterial cells have for their food source. Okay? Bacterial cells, think of, I don't know, Think of, you offer your kids broccoli, you offer them ice cream. What are they going to use first? Most kids will eat the ice cream first. Okay? They will. Bacteria have that type of relationship with glucose. Given the choice of glucose and lactose, if glucose is present, bacteria are will metabolize the glucose before they touch the lactose. Okay? Which we see in this chart is just showing the use of the glucose before they are going to switch gears and use the lactose. This is actually going to be controlled through a process that sits, a component that sits right before the lac operon. So right before the lac operon, there is a cap binding site. Okay, and the cap binding site is the location where cyclic AMP is going to bind to promote the use of that promoter of the lac operon. Okay, so cyclic AMP, when glucose levels are high, cyclic AMP is going to be low, and therefore it can't bind CAP. When when glucose levels are low, cyclic AMP is high, so it binds CAP. Okay, so this is what the top is showing where we have, actually just right here. So cyclic AMP is when it's absent, so glucose is present, cyclic AMP is not bound. We're not going to see transcription of that, we'll see limited, I should say. It's the promoter is not as easily recognized, so limited to, so very some but not a lot of the transcription of that lac operon. But when CAP is bound, it promotes the recognition of the promoter, and so that we get that lac operon produced. If lactose is not present, the repressor prevents the lac operon from being transcribed. Okay, so you kind of have two parts to the equation. For optimum production and transcription of the lac operon, glucose must be absent, lactose must be present. Okay? Again, because of that preferential to the metabolism of glucose prior to lactose. So if lactose is in high concentration, even if lactose is in high concentration, if glucose is present, you'll have minimal, the repressor will be gone from the lac operon, but you'll have minimal potential for that promoter to be recognized. There's less chance that promoter to be recognized and our products for metabolism of lactose to happen. You might get some, okay? There's a leaky process. You might get some, but you're not going to get full induction until glucose levels are low to absent. Okay, once glucose levels are low to absent, that's when you're going to get full induction, and it'll switch gears to utilize that lactose as an energy source. All right. How am I at today, slide-wise? Okay. We'll go ahead and this stops this. Next slide introduces into the mutations, so we'll go ahead and stop here for today, because this is a new concept. The operons is a new concept for you guys to digest. If you have a lab quiz that you need to if you have a lab quiz that you need to take,
That's where you get me.
not retake, but to take, because you missed it.
Thank you.
Then you can, you're going to want to stay. Okay? If you don't have